Elearning Jamaica Limited, in association with the Ministry of Education and Czec Jamaica Limited, present a series of e-learning videos geared towards preparing students for CXC examinations. This series focuses on biology. There are 10 videos, each lasting 20 minutes. Your narrator, Michael Anthony Cuff.
This video focuses on Section B of your syllabus, which covers life processes. Learning outcomes. At the end of this section, you will be able to explain what an enzyme is,
Yeah, would be good. I mean, to go along with the orange juice.
Have you ever wondered what causes peeled bananas and yam to change color when left exposed to air? Certain ground provisions contain an enzyme called polyphenol oxidase. When peeled or exposed to oxygen, the enzyme initiates a chemical reaction which results in a brown color being observed. However, when an acid is added to the exposed area, the chemical reaction is slowed. This is the reason why you may observe Jamaicans adding vinegar or lime juice to water when they are cooking ground provisions. Sliced apples left exposed to air over a period of time also turns brown and lose attractiveness. The same chemical reaction that was mentioned in the previous shot also occurs in certain fruits. People are becoming more health conscious. As such, more fruits are being consumed now than before. On a daily basis, we observe fruit vendors preparing fruit plates for people on the go. If you notice, the peeled fruits are placed in a sealed container and kept refrigerated. This procedure not only prevents the entry of microorganisms, but also reduces the chemical reaction or browning process that occurs upon the exposure of the peeled fruits, in particular apples, to air. Think for a moment, would you purchase or partake of a fruit salad that looks unappealing? Enzymes are also present in products used on a daily basis to remove stains. Stains are discolorations caused by the chemical or physical interaction of two dissimilar materials. There are different categories of stains, some of which are oil-based and or protein-based. Sweat is a protein-based stain. Detergents contain enzymes which removes stains. Here's how it works. Molecules that make up the stains form a complex with the enzyme. This results in the enzyme breaking the stain molecules into smaller sections that may be more easily dissolved. So far, the term enzyme has been used to describe a substance that is involved in chemical reactions. Let us get deeper into how these substances actually work. Enzymes are proteins made by living cells. In other words, they are biological catalysts which speed up chemical reaction without being changed in the process. Each enzyme has an active site which is formed due to the folding of long chains of amino acids. Another molecule, called a substrate, binds to the active site of the enzyme to form an enzyme-substrate complex. The substrate is the substance that the enzyme acts on in a biochemical reaction. This occurs so that a reaction can take place. When the reaction is complete, the product or products leave the active site. Without these catalysts, reactions would occur too slow to sustain life. One such reaction is digestion. When we eat, the food is broken down into simpler forms so that it can be absorbed and transported by the blood to cells for use. There are different types of digestive enzymes. Enzymes that break down carbohydrates are called carbohydrases. For example, amylase, which breaks down starch to glucose. Enzymes that break down proteins are called proteases. For example, trypsin, which breaks down proteins into smaller units called polypeptides. Enzymes that break down fats are called lipases. For example, lipase, which breaks down fats into smaller units called fatty acid and glycerol. Let's review. Enzymes are biological catalysts which speed up chemical reaction without being changed. Carbohydrases break down carbohydrates. Proteases break down proteins. Lipases break down fats. Learning outcomes. At the end of this section, you will be able to explain how enzymes work in living organisms. List the properties of enzymes. State the effect of temperature and pH on enzyme action. How do enzymes work? Scientists have come up with two hypotheses to show how enzymes work. One is called the lock and key hypothesis, and the other is called the induced fit hypothesis. In the lock and key hypothesis, the enzyme is like a lock and the substrate like a key. The key, which is the substrate, has a shape that allows it to fit directly into the active site of the enzyme. So the substrate fits into the active site without any change in the shape of the enzyme. For example, acrosomal hydrolytic enzymes which allow for sperm penetration into the egg. This is a very specific lock and key so that a species can only fertilize its own eggs. With the induced fit hypothesis, however, the active site of the enzyme changes slightly to accommodate the substrate. In order for many reactions to occur, substrates need to be given extra energy in order to be converted to a product. This additional energy is referred to as activation energy. Enzymes allow reactions to occur faster by reducing the activation energy needed for the reaction to occur between a substrate and a product. For example, a teenage boy is interested in a girl
There's this girl, right?
and uses various approaches to get her attention.
I bought her roses. I've complimented.
But each time, he is unsuccessful in his
I've
attempts.
done everything, and she's just not giving me the time of day.
However, a mutual friend of both parties, one who the girl trusts and confides in, assists the boy by putting in some good words to the girl on his behalf.
You know Adrian, right? Yes, the one that buy you the roses, the chocolates, all of that. Like you, he's a good person, he has good grades, he's a peer counselor, he's a prefect, what more you want? Right?
These words give the teenage boy the necessary push over what initially was a barrier. The end result is that the boy and the girl starts a relationship. In this analogy, the mutual friend is the enzyme. The friend enzyme lowers the barriers that normally prevented chemical reactions from occurring or slow them down by decreasing the required activation energy. Thus, in the presence of enzymes, reactions proceed at a faster rate. Furthermore, the enzyme is not consumed or altered by the reaction. A graph can be used to show how enzymes reduce activation energy. Enzymes act as builders and breakers. Anabolism includes the chemical reactions that cause different molecules to combine to form longer, more complex ones. Catabolism includes the chemical reactions that break down complex molecules into simpler ones. For example, when we fast and our body is low on energy, special enzymes break down stored glycogen, fat and/or protein molecules so that our cells can obtain energy. Let's review. 2 hypotheses to show how enzymes work are the lock and key hypothesis, the induced fit hypothesis. Activation energy is the additional energy that is needed to convert substrates to products. Enzymes allow reaction to occur faster by reducing the activation energy. Learning outcomes. At the end of this section, you will be able to list the properties of enzymes. State the effect of temperature and pH on enzyme action. Identify practical uses of enzymes. Enzymes have the following properties. They are all proteins. They are specific to particular reactions. They are inactivated by high temperatures. They are sensitive to pH. They are inhibited by poisons. Temperature and enzyme activity. Enzymes work best at an optimum temperature. At low temperatures, the enzyme is inactive. However, as the temperature increases, more kinetic energy is provided to the molecules involved. As such, the numbers of collisions between enzyme and substrate will increase. Enzymes are denatured above the optimum temperature. This occurs because the bonds which holds the protein structure breaks and causes the active site to lose its shape. Therefore, a reaction does not occur. The normal body temperature of humans is 37 degrees Celsius. Digestive enzymes work best at this temperature. In our saliva, there is an enzyme called amylase, which converts starch to maltose. This is why, as we chew bread or crackers, there is a sweet taste prior to swallowing. Explanation of practical activity. An enzyme called polyphenol oxidase is present in bananas, which results in brown pigments when they are exposed to oxygen. However, boiling stops the action at that enzyme. pH and enzyme activity. pH is the measure of the concentration of hydrogen ions in a solution. The lower the pH, the higher the hydrogen ion concentration. Hydrogen ions can interact with amino acid side chains and, as such, affect the way in which they bond to each other. Enzymes work best at an optimum pH. A pH which varies from the optimum pH can cause the enzyme to denature. Catalase is an enzyme found in most tissues such as liver, potato, and peas. Catalase is one of the fastest enzymes known. It catalyzes the breakdown of hydrogen peroxide into water and oxygen. Hydrogen peroxide is made naturally in cells and, if allowed to accumulate, becomes toxic. Mild acids such as citric acid and/or acetic acid is oftentimes placed on fruits and ground provisions to prevent discoloration. This is very important in industry, especially since consumers tend to think that browning means that the produce is spoiled. Do you recall the young man refusing to eat the apple because they were unattractive at the beginning of this presentation? Let's view the effect that orange juice would have on the apple slices. The final property of enzymes that we will focus on is how they are affected by poisons. Enzymes are inhibited by poisons. Poisons can bind to the enzymes and cause distortion of the enzyme's active site. Thus, the enzyme is unable to attach itself to the substrate and carry out its function.
Snake venom can be broken down into three main groups. This would include those that cause tissue necrosis or tissue death, right? Those that affect the coagulation system or the animal's ability to clot whenever they're bleeding. And thirdly, affects the musculoskeletal system. That means that this venom that causes paralysis. Pit vipers, which would include like your rattlesnakes and these kind of snakes, their venom tend to be more or less cytotoxic, right? They are composed of primarily a phospholipase A-type enzyme, as well as one called hyaluronidase. And these enzymes break down tissue, causing massive cell death. And this is one of the ways that they would cause injury. So typically when an animal is bitten by one of these snakes, the local area where the bite occurs starts to swell, becomes very red. With time it becomes gangrenous because the cells are dying. Secondly, for the cobra, for example, the venom more or less has an effect on the coagulation system. But whenever these snakes bite you, your coagulation system doesn't work properly, and hence you tend to bleed out, right? Thirdly, the group of snakes that would affect the paralytic system tend to be limited to, like your coral snakes, for example. These tend to be more common in the States and also in South America. And in Trinidad, for example, one of our neighbors also has the coral snakes. Now, these snakes, however, tend to affect, as I said, cause paralysis. And they do this because certain enzymes, they bind to certain receptors on your muscle fibers and cause them to continue to twitch. And therefore, you suffer paralysis. It primarily affects the respiratory system. Hence, you get respiratory paralysis and the animal dies because it cannot breathe.
Enzymes in industry. The use of enzymes in industry has become very important. In industry, enzymes are used for many processes such as baking bread and making dairy products. Protease and lipase enzymes are also added to washing detergents to remove stains and grease. Some bacteria secrete enzymes that are useful to humans, hence in food production. Protease enzymes are also used as meat tenderizers. Papaya contains a protease called papain, which makes meat tender. Let's review. Enzymes work best at an optimum pH. A pH which varies from the optimum pH can cause the enzyme to denature. In industry, enzymes are used for many processes such as baking bread and making dairy products.